Hello, I'm Dr. Stephen Pipe from the University of Michigan, and I had the opportunity to present the first data from the phase 3 HOPE-B gene therapy trial in adults with severe to moderate severe hemophilia B. Etranacogene Dezaparvovec is an AAV5 vector, and it contains the hyperactive Padua factor IX variant. So this enhances the activity of the expressed factor IX protein, and it builds on the efficacy and safety that has been demonstrated previously by this vector, which has shown stable expression of a wild-type factor IX now out 4.5 to 5 years of follow-up. A phase 2b study with this enhanced vector called AMT-061. It's now in long-term follow-up, and it's demonstrated a mean factor IX activity at 2 years of 44.2%. Now, for this phase 3 trial, notably in contrast to all other AAV-mediated liver-directed gene therapies for hemophilia to date, the pre-existing anti-AAV neutralizing antibodies were assessed but not used as an exclusion criteria. The participants were evaluated in a lead-in phase of 26 weeks. This was to assess bleeding events occurring during the standard of care continuous routine factor IX prophylaxis. And then the participants were dosed at 2 times 10 to the 13th genome copies per kilogram as a single approximately 1 hour infusion. And then we followed them afterwards. No prophylactic immunosuppression was used. Now, the study has 3 co-primary endpoints to evaluate efficacy and durability, and I'm reporting on the first of these co-primary endpoints, which was a central one-stage factor IX clotting assay at 26 weeks of follow-up. There were 54 participants in the study, ranged from as young as 19 up to age 75. More than 80% were severe phenotype, and more than half had prior hepatitis B or C infection. They were about equal prior treatments with standard half-life and extended half-life factor IX. And notably, 42.6% of the participants had neutralizing antibodies directed to the AAV5 capsid, and the max titer was over 3,200. During the lead-in phase, these 54 participants reported 123 bleeds on factor IX prophylaxis. We observed robust expression by 3 weeks post-dosing, and this measured a mean of 37.2% at week 26. And importantly, there was no observed correlation of pre-existing neutralizing antibodies with factor IX activity up to a titer of 678. There was one participant who had a titer over 3,200, and he did not respond. Now, relative to the lead-in, the total bleeds decreased by 83% and treated bleeds decreased by 91%. Importantly, 72% of the participants experienced 0 bleeds post-dosing, compared to only 30% of those during the lead-in. In the per-protocol population, 98% of the patients were able to discontinue prophylaxis and have remained prophylaxis-free, and this resulted in a 96% reduction of factor IX usage. Now, from a safety perspective, 9 patients received steroid treatment for transaminase elevations. All discontinued their steroid use prior to week 26, and the factor IX activity was preserved in the mild range. 7 patients experienced infusion-related reactions, and the infusion was discontinued in 1 patient who only received approximately 10% of the dose. All the others were able to continue or resume their infusions and were able to get the full dose. Overall, no inhibitors to factor IX were reported, and no relationship between safety and neutralizing antibodies was observed. There was 1 serious adverse event of hepatocellular carcinoma. Now, this patient had a number of risk factors for this pathology. It's been considered possibly related because of the context of this gene therapy, and this was reported after the data cut, and full analysis is going to be ongoing over the coming months. So the high-level output from this study so far, this is the first report of a phase 3 study in patients with Hem B. It's the largest gene therapy trial to report to date. Mean factor IX activity significantly increased to near normal levels at the 26-week post-dosing. No prophylactic immunosuppression. 52 of the 53 patients received a full dose and responded, and this included patients with pre-existing anti-AAV titers up to 678. All the patients were able to discontinue prophylaxis who got the full dose of treatment, and bleeding was abolished in the majority of patients throughout the 26 weeks. The most common safety findings were transaminase elevations that required steroid treatment and some infusion-related reactions, but this supports a 6-month safety profile that's really consistent with the early phase studies. So a final analysis is planned at a year, and hopefully this will be able to support market authorization applications. Thank you very much.